And this presentation is going to cover the second part of metabolism, which focuses on the role of enzymes. Metabolic pathways are actually made of enzymes. Enzymes pass along substrate molecules one to the next until you finally get your product at the end of the whole pathway. Keep in mind, though, that metabolic pathways are not always straight lines. They're almost always very connected, diverging, and converging with other metabolic pathways. So to understand them, we separate the pathway out from the others, and we look at each step along the way, and we talk about the enzymes and the products. But in real life, in real cell biology, it's a complicated mess, which somehow works together to maintain homeostasis, which of course keeps life in balance. Enzymes are three-dimensional proteins, and that means that they have a shape in 3 dimensions that's determined by the way they've been folded. And part of this folding produces a location called the active site. And the active site is shaped in a way that allows it to receive a substrate molecule. This allows enzymes to function as biological catalysts. And all that means is enzymes have the ability to make a chemical reaction that normally would happen very slowly take place very quickly in the presence of enzyme. Enzymes work best at a certain pH and a certain temperature. If you change the pH or you change the temperature, you're going to dramatically alter how quickly that enzyme works, if it still works at all. And finally, enzymes are absolutely necessary to the way our bodies and the way our cells work. Without them, life wouldn't exist. So let's watch what happens. This substrate is going to fit into the active site, and watch what happens to the shape of the enzyme molecule when it does this. You'll notice the whole thing changes. This idea is called the induced fit model, and it's based on the supposition that once the substrate and the enzyme form an enzyme-substrate complex, to reset this enzyme, to get it back to its original shape, some energy is going to have to be put in, and usually that energy is in the form of ATP. So if we phosphorylate this enzyme molecule, we allow it to reset, release the product, and take in another substrate molecule, and go over and over and over again. Enzymes are recycled millions of times because of their ability to open and close with a little bit of energy being put in each step. It's a lot like a lock and a key model, but in the case of a lock and a key, the lock here actually changes shape in response to the key being placed into it. Of course, the key is the substrate. Okay, enzymes function as catalysts because they have the ability to lower activation energy. And if you look at this diagram here, you can see that to get from reactants to products, it's a downhill. But before you can go downhill, you have to go uphill. And this uphill initial step is called the energy of activation, or E sub A. To get from reactants to products, you have to overcome the energy of activation. And nothing's going to happen until the reactants get pushed uphill a little bit. And the energy of activation without an enzyme is much higher than the energy of activation with an enzyme. I like to think of it as being like striking a match. The chemicals on this match head are going to burn and convert this wood into carbon dioxide and ashes pretty quickly. But they're not going to do it sitting on the table. They have to be elevated or lifted uphill by rubbing it on the strike surface. And this, of course, adds enough heat, which is a form of energy, to allow the reaction to get started. Enzymes lower activation energy so that the reaction will happen with less energy input needed. The enzyme's active site structure is going to allow it to place stress on the substrate molecule in a way that makes it change. And in the case of this pathway here, we have an enzyme called sucrase, which is going to interact with a disaccharide sugar called sucrose and change sucrose into products, glucose and fructose. So you can see this is a catabolic process because we're going to be breaking a bond. Now, sucrose will naturally break down into glucose and fructose, but it takes a very long time because sucrose is a very stable molecule. Now, if your body needs to digest sucrose into glucose, it uses an enzyme like sucrase. And sucrase, its three-dimensional structure, has evolved over time to allow it to receive a sucrase molecule and hold it in a way that it places stress on this covalent bond and breaks it, producing glucose and fructose. Again, refer back to that induced fit diagram we looked at earlier, and it'll show you how the molecule shape changes, or the enzyme shape changes, in order to cause the substrate to change into the product. Another good example of this, I remember being told years ago, is that it would take you an enormously long period of time to digest everything in this hamburger if you didn't have enzymes and if your body temperature was at room temperature. But with enzymes and at 98.6 degrees, it only takes about 8 hours to digest everything in this hamburger, especially the proteins, which take the longest. Enzymes, of course, also regulate reactions. They can be places where the system can either speed up or slow down chemical reactions. And one of the simplest ways of regulating an enzyme pathway is something called competitive inhibition. And all this means is the enzyme's active site is going to be gunked up or clogged up by an inhibitor. I like to think of it as putting chewing gum in the lock. So in this diagram, if this purple material gets into the active site before the substrate does, you can't get any product. Pretty straightforward there. Many of the painkiller drugs that you can buy at the drugstore are actually competitive inhibitors. They're going to lock into the active sites of receptor proteins on nerve cells, and this makes the brain unable to receive signals, for example, pain signals. And this is going to last until these receptor proteins are cleared or rebuilt. More regulation. There isn't just competitive inhibition. There's a non-competitive inhibition process that's possible here. And what that means is the enzymes, of course, have active sites that a substrate can fit in, but they also have another site which is called the allosteric site. And the allosteric site can be activated or deactivated by something being put into it. So, for example, if the allosteric enzyme has to be switched on, that means that the allosteric site has to receive the activator to turn it on. And you can see here if you look closely, the substrate can't fit into the active site unless the allosteric activator is bonded to it. And once that happens, its shape changes or its conformation changes, induced fit model, remember, so that the substrate can then fit onto the active site and change into the product. Non-competitive inhibition is involving allosterism again. You have an additional site. So here's the substrate indicated in black here, and we have a gray enzyme. And here we have an allosteric site. Allosteric just means a different location, different from the active site. And you can see that the enzyme can be shut down in this case because an inhibitor fits into the allosteric site. And when the allosteric site receives the inhibitor, the active site now no longer works. So I'm hoping you can see that allosteric enzymes can either be switched on by an activator or switched off by an inhibitor at a different site, a site different from the active site where the substrate is going to fit. All right, feedback inhibition. This is actually kind of wonderful. I kind of like this. This is when a product made by pathways acts as an inhibitor on that very same pathway. It's efficient because the pathway regulates itself. So let's look at it this way. You have the substrate fitting into enzyme 1, producing an intermediate which fits into enzyme 2. The intermediate makes a product, and the product fits into the active site of the original enzyme and prevents it from receiving more of the original substrate. So it's a process that shuts itself down. So let's use our diagram from earlier to see this. So here's our enzyme pathway, and we're getting down towards our final product. Okay, and what happens here? Let me get this. What happens is the final product is going to become a competitive inhibitor for enzyme 1. So once you get enough final product made, it starts shutting down enzyme 1. If enzyme 1 is shut down, then nothing's made to run enzyme 2, and the whole pathway shuts itself down. So feedback inhibition is a great way of shutting down a metabolic pathway when enough final product exists. So when the concentration of the final product gets high enough, the system shuts itself down, and when the final product concentration falls, the system turns itself back on. It's a self-regulating feedback loop, and I think that's kind of nice. Let me get rid of this now. All right. Okay, that's the end of our slideshow, and I hope it helps you understand